Welcome back everyone. In this video, I'll be talking about the lipofuscin of the retinal pigment epithelium. Actually, I'm going to read this topic from the book Fundus Auto Fluorescence by J V Forrester. So, second chapter, lipofuscin of the retinal pigment epithelium. Page number 14. Lipofuscin accumulation is strongly associated with retinal aging and the progression of age-related macular degeneration (AMD). Such lipofuscin accumulation is typical of highly metabolic post-mitotic cells in a variety of aging mammalian tissues, for example liver, heart, brain, testes, and retina, that accumulate non-degradable lipid protein aggregates within lysosomes. In the retinal pigment epithelium (RPE), lipofuscin can be derived from autophagy, degradation of spent intracellular organelles such as mitochondria, Golgi, and endoplasmic reticulum. Autophagy means self-eating. Cell eats its own contents for the renewal of the contents. Or the ingestion of photoreceptor outer segments. The failure of the RPE lysosomal system to completely degrade this material is probably the result of peroxidative modifications. The high level of lipofuscin in the RPE leads to cellular dysfunction, which in turn contributes to photoreceptor dysfunction and degeneration. Retinal pigment epithelium lipofuscin. Retinal pigment epithelium lipofuscin granules accumulate in the central to basal portion of the cell as spherical membrane-bound granule 1 to 2 micrometer in diameter. In addition, in aged donors, lipofuscin can be observed complexed with melanin-forming aggregates of heterogeneous composition and size, in which case they are termed melanolipofuscin. Ex vivo and in vivo analysis demonstrate an age-related increase in lipofuscin content up until the age of 70 years. Thereafter, lipofuscin levels plateau or even decline. The reason for this decline is not clear, but it may involve one or a combination of lipofuscin degradation routes. For example, voiding into the extracellular space as basal laminar deposits or precursor material to drusen, or association with melanosomes. Concomitant with the accumulation of lipofuscin granules are a decrease in melanosomes and an increase in melanolipofuscin granules. On the basis of early morphometric data, the accumulation of RPE lipofuscin was erroneously considered to be bimodal, but it is now apparent that such accumulation is linear, at least up to until the later decades, when the effects of age-related pathology begin to have an impact. Lipofuscin demonstrates a regional distribution across the fundus. The distribution of lipofuscin demonstrates a retinal topography similar to that of rod photoreceptors, suggesting that ingested POS are a major substrate for RPE lipofuscin. Alternatively, the highest density of photoreceptors will require the highest level of metabolic activity for the retinal pigment epithelium, and thus mitochondrial turnover and autophagy will be much higher. The cytoplasmic volume occupied by lipofuscin increases significantly with age. Feeney, Bones, and colleagues demonstrated that 8% of the cytoplasmic volume of the central RPE is occupied by lipofuscin granules by 40 years of age, and this increases to 19% by 80 years of age. It is likely that this would affect metabolic function, intracellular compartmentalizations of organelles, and lysosomal activity. Now there is a figure 2.1. Micrographs showing the accumulation of lipofuscin in human RPE cells. Transmission electron micrograph of human RPE from a 52-year-old donor reproduced courtesy of John Marshall, St. Thomas Hospital, London. Note: photoreceptor outer segment POS melanosome M located toward the apical portion of the cell, lipofuscin granules L in the central to basal region, a high density of basally located mitochondria MT, Brooks membrane BM Fluorescence microscopy of tissue sections from 43 and 97-year-old donors showing an increase in fluorescent lipofuscin granules with increasing age. C. A scanning electron micrograph showing RPE cells from an 85-year-old donor. The cells are so packed with lipofuscin granules that their outline can be seen tracing against the cell membrane. Reproduced courtesy of John Marshall St. Thomas Hospital, London. D. A confocal image of flat mount human RPE showing the variable distribution of lipofuscin between individual cells. The annulus devoid of lipofuscin surrounds a roots. Figure 2.2, a graph showing a number of lipofuscin granules per micrometer of Brooks membrane. For non-age-related macular degeneration donors and donors with AMD as a function of age. Regression analysis showed a significant increase in RPE lipofuscin granules up to the age of 50 and a tendency to plateau between 60 and 100 years. So on x-axis, age in years 0, 25, 50, 75, 100, and lipofuscin number per Brooks membrane length in micrometer 0.0, 0.02, 7.5, 2.55 and 7.5. So red line, red dots, there is a blue line and triangles, blue and green triangles. Non-AMD less than 60, p-value less than 0.0001, non-AMD more than 60, p-value 0.0882, AMD p is equal to 0.5062. Reducing the functional capacity of the RPE with increasing age. In old age, the cytoplasmic volume occupied by the equatorial and peripheral RPE is considerably less than in the central retina. In addition to the regional distribution of lipofuscin across the fundus, there is considerable cell-to-cell heterogeneity in lipofuscin density in any particular region of the retina. It is evident that two adjacent cells, presumably with the same functional and metabolic load, can have high, intermediate, and minimal levels of lipofuscin. This would support the proposal by Burke and H. Gelmeland, I don't know the pronunciation of these names, weird names, that mosaicism of the RPE is a natural result of normal mechanisms regulating gene expression during development and postnatal aging. This heterogeneity may help explain the highly focal nature of the lesions associated with pathologies such as AMD. Among the different tissue lipofuscins, RPE lipofuscin has a number of unique properties that determine its photophysical properties and its potential for contributing to RPE dysfunction, as will be discussed in more detail below. First, RPE lipofuscin is derived at least in part from POS, photoreceptor outer segment, POS, and thus contains significant quantities of retinoids, including the diretinal conjugate A2E. Second, RPE lipofuscin is a broadband absorber that is constantly exposed to visible light and is located in a high oxygen environment, which makes it an ideal substrate for retinal photodamage. Lipofuscin genesis and composition. Although RPE lipofuscin is believed to result from the incomplete degradation of photoreceptor outer segment, and photoreceptors means rods and cones, outer segment. They do have outer segment, inner segment, and the nuclear portion and synaptic endings. So, although RPA lipofuscin is believed to result from the incomplete degradation of the photoreceptor, that is for rods and cones, outer segment, POS is photoreceptor outer segment, and autophagy of spent or damaged intracellular organelles, and relative contributions of these pathways remain unknown. It is thought that prior oxidative damage to these substrates renders them undegradable by the lysosomal system. The universal intracellular degradation machinery that contains over 40 hydrolytic enzymes. However, prior oxidation does not account for the fluorescent properties of lipofuscin. Singh, Eldred, and Katz showed that the autofluorescent products of lipid peroxidation differ from those of lipofuscin. Although we have gained an understanding of the initial substrates involved in lipofuscin genesis, we still have a limited understanding of composition of the nature of lipofuscin granules. Probably the first attempt at analysis was made by Eldred and Katz. I don't know the pronunciation of these names. These are like some new names for me to pronounce them correctly. Who used Folk's extraction to separate lipofuscin into three fractions: chloroform soluble, methanol soluble, and insoluble. Thin layer chromatography analysis of the chloroform fraction revealed 10 discrete fluorophores, 2 green emitting, which co-migrated and retinal palmitate, 3 emitting yellow-green, and 1 golden-yellow, and 4 orange-red fluorophores, the most prominent of which was subsequently identified as A2E. Further studies by other researchers identified the fluorophores as retinoid derivatives. This was followed by many studies on the role of A2E, the only readily synthetic component of lipofuscin, and demonstrations of its phototoxic and lysomotrophic properties. However, the content of the bulk, more than 70% of lipofuscin material, which is located at the insoluble interface on Folk's extraction, has largely been ignored. It has long been considered that lipofuscin consists of cross-linked oxidatively modified lipids, proteins, and sugars, with the proteins content ranging from 10% to 70% of the lipofuscin granule. The first proteome for RP lipofuscin was reported by Schutt and colleagues in 2002. They identified over 65 abundant cellular proteins, including cytoskeleton proteins, proteins of transduction, enzymes of metabolism, proteins of mitochondrial respiratory chain, ion channel proteins, and chaperones, which supported the origin of lipofuscin as a combination of photoreceptor, phagocytosis, and autophagy. Furthermore, analysis of the samples revealed that many of these proteins were damaged by aberrant covalent modification of MDA, 4-HNE, and AGEs, advanced glycation end products. In a subsequent proteome analysis of RP lipofuscin, Warburton et al. Identified 41 cellular proteins. Surprisingly, only 11% these proteins had been identified in the proteomic analysis of Schutt and colleagues. Although there are numerous explanations for this discrepancy, the likely problem lies in the purity of the starting material. Lipofuscin is generally isolated by repeated sucrose density gradient centrifugation. However, there is usually some residual contamination by cellular debris. While investigating this further, we demonstrated that highly purified debris-free RP lipofuscin granule contains little or no protein, but does contain significant amounts of modified material, such as carboxyethyl pyrrole adducts. In conclusion, although our understanding of the composition of lipofuscin in the RPE remains limited, it does appear that protein makes only a very small contribution, if any, and that oxidized and modified lipids from the outer segment and mitochondrial membranes may be major contributors. Figure 2.3, lipofuscin composition and photoreactivity. A photograph of lipofuscin A after Folk's extraction. The top layer is the methanol water phase. The bottom layer is the chloroform soluble phase, and insoluble material is located at the interface. B. Comparison of the action spectra of initial rates of photoinduced oxygen uptake in suspensions of DMPC liposomes containing both the chloroform soluble and insoluble triangle and inverted triangle respectively interfacial material plus a combination reconstituted lipofuscin. A square, a diamond square. The arithmetic addition plus is shown of the rates measured separately for the chloroform soluble and chloroform insoluble fractions. The inset shows the change in content of dry mass of the chloroform soluble and chloroform insoluble material extracted from the lipofuscin granules as a function of donor H, horizontal bars ST of the donors of the pooled samples, vertical bars ST of the dry mass measurements reproduced courtesy of investigative ophthalmology of the visual science from reference 18. C. Comparison of the thin layer chromatography profile of the chloroform soluble fraction of pooled lipofuscin granules from donors of different ages. 1. 50 to 59 years, 2. 60 to 69 years, 3. 70 to 79 years. The fractions show a number of different fluorophores when excited by ultraviolet irradiation. Spectral characteristics of RPE lipofuscin. Retinal pigment epithelium lipofuscin granules exhibit a broad and wavelength dependent absorption spectrum with a decrease in absorption toward increasing wavelength. The excitation and emission spectra of lipofuscin granules are shown in figure 2.4 and a summary of excitation and emission peaks is provided in table 2.1. RPE lipofuscin granules typically exhibit four main regions of interest in the emission spectra when excited at 364 nanometer. The main peak located at 600 to 610 nanometer, a blue-green shoulder located at 470 nanometer, a green-yellow shoulder at 550 nanometer, and in case of lipofuscin from individuals over 50 years, a far red shoulder at 680 nanometer. Similar emission peaks are observed at 500, 610 and 680 nanometer when lipofuscin is excited at 476 nanometer, which is similar to the excitation employed by in vivo autofluorescence system. More than or equal to 488 nanometer. Excitation spectra with emissions monitored at 610 nanometer or 570 nanometer show a main excitation peak at 470 nanometer with two subsidiary peaks at 370 and 405 nanometer. Time resolved fluorescence microscopy has identified 4 decay components in lipofuscin with decay times of approximately 0.21, 0.65, 1.75 and 6.7 nanosecond. Wavelength resolved decay analysis demonstrated that 3 main fluorescent components of lipofuscin do not strongly interact and thus appear to be excited directly. The spectral characteristics of melanolipofuscin are intermediate between those of lipofuscin and melanin. The fluorescence properties of lipofuscin granules exhibit age-related changes. The fluorescence intensity of lipofuscin granules increases by up to 40% with increasing age. Whether this is due to increased deposition of material in the lipofuscin or a change in a composition is unclear. It should also be emphasized that there is considerable heterogeneity in the fluorescence properties between individual lipofuscin granules with variations in both emission maxima and spectral shape. Thus, lipofuscin spectra generally represent an average of all these spectral differences. So that was about figure 0.4. Figure 2.4. Excitation and emission spectra of intact lipofuscin granules isolated from donors of different ages. Excitation spectra were monitored with emission performed at 570 nanometer and emission spectra were monitored and excitation performed at 364 nanometer. Lipofuscin granules were prepared from different age groups: A. 5 to 29 years, B. 30 to 49 years, and C. more than 50 years. Excitation and emission spectra are expressed in arbitrary units, AU. Summary of the main fluorescence peaks at lipofuscin granules from different age groups observed in figure 2.4. Excitation spectra were monitored with emission performed at 570 nanometer and emission spectra were monitored with excitation performed at 364 nanometer. Sample age in year, then excitation in nanometer, emission in nanometer. Total peak amplitude. So sample age 5 to 29, excitation 370, 404, 470 nanometer, emission 470, 550 and 600 nanometer, almost 50 to 100 nanometer difference. And sample age 30 to 49, excitation in nanometer 370, 405, 470, emission again 470, 550, 600, same nanometer. Then sample age in years 50 to 80, then excitation wavelength in nanometer 370 to 470 nanometer, then emission wavelength in nanometer 570, 600 and 680 nanometer. Total peak amplitude 1, 1.71, 1.95 respectively for these age groups. Aging lipofuscin and the potential for chronic light damage. The spectral characteristics of lipofuscin, together with its localization in a high oxygen environment and diurnal exposure to visible light, make it an ideal substrate for photochemical reactions. Our laboratory was the first to demonstrate that lipofuscin is able to photo-generate a reactive oxygen species, ROS, when we demonstrated that exposure of isolated human RPE lipofuscin granules to light results in the generation of the superoxide anion. That study also showed that the photocoagulation of reactive oxygen species was dependent on both light intensity and wavelength, with the highest levels of superoxide anions being generated at higher visible light intensities and shorter blue wavelengths of visible light. Subsequent studies demonstrated that isolated human lipofuscin granules are able to photogenerate significant quantities of singlet oxygen, hydroxyl radical, hydrogen peroxide, and the longer-lived lipid peroxides. There is evidence that the overall photoreactivity of lipofuscin increases with increasing age. It appears that this is due to an increase in one or more of the components in the chloroform insoluble fraction obtained from lipofuscin granules. Analysis figure 2.3 analysis of blue light photoreactivity in isolated RPE cells from donors of different ages demonstrated a significant age-related increase in oxygen photouptake and indicated of reactive oxygen species formation, primarily due to the presence of lipofuscin within the cells. Not surprisingly, the lipofuscin photo-generated reactive oxygen species are capable of eliciting oxidative modification to lipids, proteins, and nucleic acids. Lipofuscin granules exposed to light induce oxidation of both intra and extragranular lipids and inactivate critical RPE enzymes, including the lysosomal and antioxidant enzymes. The impact of these photooxidative reactions on cell function was assessed in a cell culture system in which lipofuscin containing RPE cultures were exposed to 2.8 meter WCM square, I think it is watt centimeter square, blue light 390 to 550 nanometer or amber light 550 to 700 nanometer or maintained in the dark. Exposure of lipofuscin containing cells to blue light resulted in lipid peroxidation, formation of the aldehydes MDA and 4-HNE, protein carbonyl formation indicative of protein oxidation, nuclear but not mitochondrial DNA damage, loss of lysosomal integrity, cytoplasmic vacuolation, and membrane bleeding, culminating in cell death within 48 hours. RPE dysfunction could be observed within 3 hours of exposure of lipofuscin containing cells to blue light and was associated with DNA damage, a reduction in antioxidant status, and a decrease in lysosomal enzyme activity. Neither cells exposed to equivalent irradiant energy, amber light, nor cells maintained in the dark exhibited any oxidative changes or loss of cell function. The ability of lipofuscin to cause photooxidative damage to all components extrinsic to the lysosomal compartment indicates the importance of longer-lived and less reactive oxygen species such as lipid peroxidase that have a greater chance to cause oxidative damage throughout the cell. The most studied of the potential photosensitizers of RPE lipofuscin is A2E. Although A2E makes a major contribution to lipofuscin fluorescence by serving as an energy acceptor from other blue light absorbing molecules within lipofuscin, it is itself only weakly photoreactive compared to the hydrophobic components of lipofuscin. Despite its weak photoreactivity, A2E can, at high concentrations, elicit blue light induced RPE cell dysfunction and cell death in cell culture. However, other A2E related molecules such as A2E epoxide are also present in lipofuscin, and these may be more photoreactive than A2E itself. Based on the spectral characteristics of photoreactivity of lipofuscin, there is a strong evidence to suggest that lipofuscin may contribute to the pathogenesis of AMD, age-related macular degeneration. First, longitudinal studies of in vivo fluorescence have shown that areas of retina with the highest lipofuscin levels are the most susceptible to degeneration. Second, the chronology of RPE lipofuscin accumulation is coincident with development of AMD. Third, since the RPE is composed of post-mitotic cells, reactive oxygen species damage will accumulate throughout the life. Fortunately, the crystalline lens attenuates the shorter wavelengths of light reaching the retina, and this effect will be greater in older luminescent lenses. However, even when the age-related changes in the transmission characteristics of the ocular media are taken into account, the overall phototoxicity of lipofuscin increases substantially with age because the protective effects of the lens senescence are partly offset by a substantial increase in the concentration of photoreactive lipofuscin granules in the retina. Although this phototoxicity increases steadily with age, the potential for photoinducible damage will be many times greater where there are focal hotspots of lipofuscin. Figure 2.5. The photoreactivity of retinal pigment epithelial lipofuscin fade RPE cells, a red hollow square and red dense square, and cells lacking lipofuscin, hollow circle and full red circle, were exposed to 2.8 meter per 2.8 meter per watt centimeter square. I think this W means watt light in 390 to 550 nanometer red square and red circle which are hollow. Then B 550 800 nanometer are maintained in the dark, the square and circle, red square and circle which are dense full, for up to 48 hours. The ability of the RPE cells to reduce MTT to a blue formation product, absorbance measured at 590 or 630 nanometer, was used as a measure of cell variability. The insets show light micrographs of lipofuscin fade RPE cells exposed to light A or maintained in the dark B for 48 hours. Phototoxicity action spectra for 3 different age groups in C are shown. So here on the x-axis there is a wavelength in nanometer, 300, 400, 500, 600, and on the y-axis you will see ranging from 0 to 30 there is the relative phototoxicity. That is figure C, and in figure A you will see the on x-axis there is a time in hours, 0, 12, 24, 36, 48, and on y-axis there is a viability as a percentage of control. Zero, 25, 50, 75, and 100. Then in figure B, you will see again the time in hours and same viability as a percentage of control. A2E and lipofuscin. Eldred and Lasky were the first to identify the major fluorophore in the chloroform soluble extract of lipofuscin as A2E. The formation of A2E begins in the POS, that is photoreceptor outer segment, with condensation reactions between phosphatidyl ethanolamine PE and two molecules of all-trans-retinal ATR released from activated rhodopsin. The product of the initial chief base reaction between a single ATR and PE is N-retinylidine PE, NRPE. The biosynthetic pathway continues through a number of steps, including the addition of the second ATR to form the phosphatidyl pyridinium bisretinoid that is immediate precursor to A2E. That is the immediate precursor to A2E, and the fluorescent pigment that accumulates in POS, photoreceptor outer segment. Although there is general agreement that A2PE undergoes enzymatic cleavage to form A2E, it is unclear whether this cleavage occurs in the outer segment prior to ingestion or after uptake into secondary lysosomes. However, A2E ends up within the lysosomes containing lipofuscin granules, and because of its non-degradable qualities, its overall concentration in the RPE not unexpectedly increases in parallel with lipofuscin accumulation. It is also evident that A2E can undergo a variety of changes, including photoisomerization and oxidation, to produce a variety of A2E derivatives. Although A2E has been shown to be present in lipofuscin, and exposure of RPE cell cultures to chemically synthesized A2E in solution can cause cell dysfunction and death, the actual importance of A2E to the overall composition and cytotoxicity of lipofuscin has never really been proven, and a number of contraindications can be found in the literature. First, the absorption properties of A2E account for only a small proportion of the visible light absorbed by lipofuscin. Second, it is unclear to what extent A2E contributes to fluorescence spectrum of lipofuscin, since intracellular A2E has a maximum emission of 560 to 575 nanometer, and lipofuscin granules have maximum emissions at 600 to 610 nanometer. Third, low concentrations of A2E protect against UV-induced DNA damage in RP cells in vitro. Fourth, A2E is not the major photosensitizer responsible for the generation of reactive oxygen species by mature lipofuscin granules. Fifth, the bioavailability of A2E in the RPE remains difficult to determine, and to date there is no evidence that A2E or its derivatives can be released from lipofuscin granules once it has been sequestered. Notwithstanding these observations, it is known that synthetic A2E applied directly to RPE cells in culture is phototoxic when exposed to the blue light. It can cause oxidation of cellular constituents, though the photogeneration of singlet oxygen. It upregulates the expression of angiogenic factors such as VEGF. It is lysosomotrophic and able to destabilize lysosomal membranes, resulting in reduced lysosomal enzyme activity, and it can reduce mitochondrial capacity, leading to apoptosis. In addition, photooxidation products of A2E can lead to complement activation. Thus, it is clear that A2E has the potential to cause RPE cell damage, but whether in vivo this occurs pre or post incorporation into lipofuscin granules remains to be shown. Association between lipofuscin and retinal degeneration. Elevated lipofuscin levels, as demonstrated by fundus autofluorescence imaging, are associated with a variety of retinal degenerations. See fundus autofluorescence in the diseased eye condition section. Although all of these conditions demonstrate increased autofluorescence and elevated numbers of lipofuscin granules within the RPE, The composition of the granules may well vary significantly. Furthermore, it is difficult to determine whether lipofuscin is a cause or consequence of these conditions. However, in the case of AMD, age-related macular degeneration, there is a considerable circumstantial evidence linking lipofuscin with the etiology of the disease. First, lipofuscin can be observed in early drusen, and these may serve as an immunogenic stimulus for the localized activation of dendritic cells and deposition of inflammatory factors. Confocal microscopy demonstrates two lipofuscin profiles in the overlying RPE. One presents as a ring of hypofluorescence surrounding drusen, and the other presents as a central area with decreased lipofuscin. Enjo and Bolton unpublished results. Furthermore, subretinal exocytosis of lipofuscin granules appears to be a feature of eyes more than 60 years of age and results in degradation of these granules in the extracellular sub-RP space. Second, the highest density of lipofuscin is located in the central retina, where the density of rod outer segments is at its highest and where there is preferential loss of rod cells in AMD. Third, the accumulation of lipofuscin is reported to be greater in whites than in blacks, and AMD is more prevalent among whites than among black persons. Fourth, chronic phototoxicity, lysosomotrophic damage, and occupation of cytoplasmic volume in RPE cells by lipofuscin and its constituent A2E throughout life will contribute to RPE dysfunction and put added stress on a highly metabolically active cell types. Fifth, longitudinal in vivo monitoring of fundus autofluorescence has demonstrated that the presence of focal increased autofluorescence is a risk factor for progression of AMD. Furthermore, zones of RPE exhibiting increased fundus autofluorescence signal are prone to atrophy, and these areas exhibit a variable loss of retinal sensitivity that suggests an association between RPE dysfunction and excessive accumulation of lipofuscin. Sixth, synthetic A2E, when photoactivated, can induce the expression of angiogenic factors in RPE cells. Seventh, photooxidation products of A2E can activate complement, and this may contribute to the chronic inflammation associated with AMD. Eighth, lipofuscin levels have been observed to be elevated in rodent models for AMD, age-related macular degeneration. Furthermore, in Stargardt disease, there is an abnormality in the ABCR gene that leads to buildup of lipofuscin fluorophore A2E. The predisposition for excessive lipofuscin accumulation in retinal degeneration such as AMD, and in particular its elevation during the early stages of disease progression, suggests that lipofuscin makes a significant contribution to the pathogenesis of AMD rather than simply being a consequence of the disease. Figure 2.6. A fundus A. A fundus autofluorescence image of a 74-year-old patient with visual acuity 20/40 taken in 2005 with a Heidelberg HRA-2 scanning laser ophthalmoscope. Several areas of RP atrophy are noted, with a small areas of increased autofluorescence at the borders of the larger lesions. At this time, the autofluorescence in the fovea was regular, and the patient was told that progression of this condition was imminent. B. Two years later, the patient was re-examined and the visual acuity had decreased to 20/200. Autofluorescence taken with the same instrument showed extensive geographic atrophy, including the foveal area. Reducing the impact of lipofuscin on aging and retinal degeneration. If, as the evidence above suggests, lipofuscin is cytotoxic and contributes to the pathogenesis of retinal degeneration such as age-related macular degeneration and Stargardt disease, then a strategy to remove or prevent further formation of lipofuscin would be efficacious. There are a number of paradigms to support this. A. Lipofuscin can be voided into sub-RP space where it appears to be degraded. B. Lipofuscin deposition can be decreased with appropriate modification of the lysosomal compartment. And C. Knockout or modification of retinal pigment epithelium specific proteins RPE65, a critical component of the retinoid cycle, prevents accumulation of the retinoid components of the lipofuscin. The most popular approach to date is to inhibit A2E formation by reducing retinoid turnover in the visual cycle. A2E deposition is an endpoint that can be easily measured both in vitro in cultured RP cells and in vivo using the ABCR mouse model for Stargardt disease. Isotretinoin, accutane, and the small molecule isoprenoid RPE65 antagonists reduce the formation and accumulation of A2E and other retinoid pigments in ABCR mice. ABCR negative mice. An alternative approach is to reduce A2E accumulation by reducing serum vitamin A using N in bracket 4 hydroxyphenyl bracket complete retinamide using N-retinamide. Although all of these approaches are promising, they have two major limitations. A. Targeting the retinoid cycle is likely to affect visual functions, and there is clear evidence that many of these agents slow dark adaptation and or reduce impaired electroretinogram ERG responses. And B. Although A2E fluorophores are reduced, there has been little morphometric analysis to confirm a significant reduction in the lipofuscin granules within the RPE. Fenretinide, currently in clinical trial, is the most recently developed agent aimed at reducing the retinoid cycle, but evidence suggests that this may actually promote choroidal neovascularization (CNV) in compromised eyes. Alternative approaches that are aimed primarily at attacking the non-retinoid composition of lipofuscin include reducing the accumulation of lipofuscin by either A. Preventing the formation of intralysosomal ROS reactive oxygen species which cross-link intralysosomal material using ion chelators. B. Preventing the formation of cross-linking adducts in lipofuscin by increasing the levels of intralysosomal glutathione S transferase and facilitating the degradation of lipofuscin substrates. There is no doubt that the next decade will see the development of a variety of therapeutic approaches for preventing or removing lipofuscin accumulation. Lipofuscin, the unanswered questions. In the last 2 decades, there has been a major emphasis on RPE lipofuscin research, and studies have provided invaluable information on the composition, photobiological properties, and association of lipofuscin with retinal degenerations such as AMD, age-related macular degeneration, and Stargardt disease. However, despite our increased knowledge, there are still many unanswered questions. First, to what extent does autophagy contribute to the biogenesis of RPE lipofuscin? Research would suggest that the mitochondria, which are abundant in RPE cells, are compromised in aging and AMD, and thus their turnover may be much greater. Second, if protein is only a minor component in lipofuscin, what are the non-retinoid constituents? Of interest, autofluorescent lipofuscin-like granules accumulate in long-term RPE cultures without exposure to photoreceptor outer segment or retinoids. These granules are highly photoreactive. Third, what is the importance of A2E within the lipofuscin granule? Is it inactive when sequestered into the lipofuscin granule, and can it be released? Fourth, what are the fluorophores in the lipofuscin? Clearly A2E is one, but there must be many more since A2E does not account for a large part of the excitation or emission spectra of lipofuscin granules, either in vivo or in vitro. Fifth, Is lipofuscin immunogenic, and does it release into the sub-RPE space lead to a localized inflammatory response? Question mark. Sixth, what is melanolipofuscin? Given the heterogeneous composition of these granules, they cannot be simply derived from the fusion of melanosomes and lipofuscin granules as suggested in some textbooks. Seventh, can we degrade existing lipofuscin or prevent its accumulation by pharmacological intervention? Finally, is lipofuscin a cause or a consequence of retinal degenerations? It is hoped that research over the next decade will clarify these issues and both improve our understanding of retinal pathogenesis and identify effective pharmacological treatments for diseases in which lipofuscin is prevalent.